now our next speaker is a full-time professor of biophysics at the Faculty of Health Sciences, University of Ljubljana, with a PhD in physics. She has supervised 31 PhD theses and established a biophysics of membranes course at the University of Doctoral School, led multiple Slovenian research agency projects and EU-funded initiatives recognized with scientific achievement awards, the National Zois Award and the L'Oréal UNESCO nomination for Women in Science. Ladies and gentlemen, please welcome here in Budapest, Professor Dr. Veronika Kralj Iglič.
Thank you very much. Thank you very much, Drs. Knaus and Bensi, for inviting me here, and also to Dr. Radisic and Dr. Stukel, who introduced me. And it's a great honor and a pleasure to tell something about our research here. So we go now to microscopic size. So we have very small particles, which are also, so the space and the microscopic things are quite connected. I think it's all over the world. The physics is the same, as we say. And we begin now with Luca. So we don't know how the life really started, and there is some guesses, and so there should be a main grandmother, this Luca, of us all, also not only human, but also other beings. And the anthropocentric view on life may be not so good, so we should take care also of other beings. And you see, then by the development, we came to all kind of beautiful creatures like eukaryote, and then we have bacteria and archaea, which are also amazing creatures. So that is just a cartoon. It's not that anybody ever saw this thing, but you can see here that there is some membrane, some, let's say, proposed idea of how this Luca, our great mother, looked like, and it has some membrane and something inside, which is kind of protoplasm. And then we have these modern models of biological membrane, of cellular membrane, and we have on the right from 1972 the Singer-Nicholson model, which is excellent. It's saying that we have a lipid bilayer with some bigger molecules floating inside. That's the proteins. And on the left we have also the artificial membrane, which we can synthesize artificially from synthesized lipids as well. So we can somehow get into the processes by our own, let's say, ideas and wishes. And it's not only cells, but it looks like that the cells communicate between each other on the nano-sized particles, and these nano-sized particles are parts of themselves. So if they want to communicate, they just take off a piece of them and send it to another cell. And therefore, the communication takes place, and also this way in pathological situations, let's say in infection or something like cancer, the metastases go in this way. So this is a communication between small particles taking place by even smaller particles which are nano-sized. And the issue on that, the physics of that, is composed on curvature. So it is the curvature of the membrane which makes the difference, how these particles will shed off the membrane and how they will then unite with another cell and deliver the cargo. So they are the spacecrafts of cells, the little things. And we call them extracellular vesicles or extracellular particles, because now they're not only membrane-bound ones which protect the cargo inside from enzymes and other things that can destroy them. So we were studying this for 30 years. First, we were doing theory. Then we met some people who did experiments. And here are the images of these beautiful structures. So these are the red cells you can see there. We all have them. And the red cells, in certain conditions, like On the right, you see that they become like, they get the spikes. This was due to the presence of detergents. So detergent can change the membrane because they intercalate into the outer layer and they make the bumps outwards, but also the inwards. On the left, you can see that they're having invaginations, and eventually this, on the top of these spikes, they form the buds which can shed off, and these are nano-sized. They're really small, and they can carry the cargo to other cells. So knowing these mechanisms is kind of important, and we can see here, look on the left side, you have the size of these structures. So they go from 100 nanometers to 10 micrometers. That's 100 times the size. It means, let's say, a person 100 meters high and a person 1 meter high have the same properties. They have the same shapes. And on the upper row, you have the calculated shapes. So there exist the models. We understand why they look like this. So physics can explain. It is the minimization of the energy. This is the fundamental, let's say, law of physics, that the system will always attain the shape or the configuration of the lowest possible energy. Nature is lazy. They favor lazy creatures, lazy material, whether it is down. The last one is the artificial one. So we see there is no wonder of life. It is the physical law that governs that, and we can understand it, and we can also interfere. So the first conclusion is that what we can say, that it's not some special kind of life, that some special magical, or I don't know, ideas which govern life, but it is physical laws, it is laws of nature, and we can, by knowing more and studying more, we can get to that to help things get better. Also in medicine, let's say, in particular, that we're interested. But besides these membrane enclosed ones, which we can understand quite well, there are also other types of small particles which living creatures produced. And there are also other mechanisms, and one of them is cellular death. So when the cell dies, it fragments, and these fragments as well reach other cells and influence them. So this communication is taking place within our organisms, between the organisms. Also viruses that we recently know can be very strong in this communication. And for example, this is one of these mechanisms. It's apoptosis, which makes the little particles, they're uglier. The ones before were more beautiful, smooth. These ones are more heterogeneous, they have different cargo. And then we come also to these magnificent creatures, which are called microorganisms. And these beautiful mannequins here are microalgae Phaeodactylum tricornutum. They have very fancy names as well. There are many types. And we brought a sample here. It's back there in one box. You can look at it, how they are brown. The algae are brown. And these are, in fact, probable astronauts that could survive without any money. So they're already there, I can say. Probably they are. Maybe not this type, but there are so many types. And as we are thinking about how we will get there, they also have high tech inside them, so we could learn from these creatures really a lot. Because we say they're superior creatures, but these are really amazing creatures. So they can produce flagella, for example, some of them, which can move them 400 of their size in 1 second. And they're doing them themselves. So we say we can produce silica particles. They have a factory already in there, and they can do it. And so I think these are the candidates that will be able to invade the space, probably, maybe not this type, but their cousins or somebody. And we can just ask them and please them and take care of them to make an atmosphere for us to be able to live, because they already did it. So that we can breathe this air here, it's them who do it. So they take the CO2 and they produce the oxygen. they're in the oceans, they're in the rivers, they're everywhere. And it's huge amount of them. It's just unseen majority of creatures doing their, let's say, charity work to us, and we should support them somehow, so they will be able to do it again, maybe in space, if we can do that. So that's the internal beauty of this creature. So look at the little particles on the left. It's smaller than 100 nanometers, and as we said, they're the spacecrafts or the Earthcrafts moving between the creatures here on Earth, but they could do it maybe also in space. And so you see the cartoon of the Luca, but then we see the cryo-electron microscope images of these little particles, which look quite similar as these ideas, or probably these people got the ideas there, but they have this corona of protein. And we're sorry that we don't know more about it. So there are some images about it, but there is much. Recently we have observed shedding of particles from microalgae. Oops. It turned out that numerous small particles. Okay, it was recorded apparently. I don't know. I'm sorry. So this is Tetraselmis chui. This algae has 4 flagella. You can see on the right there, the last image, and there are hairs and scales coming off from this flagella, and these are the images on the left, how they look like, these hairs and scales. And these were already shown that they deliver the cargo to the cells. They were eukaryotic cells, so they're not picky. They deliver also to human cells or other cells. And therefore, we are performing research. But we can see also now other, let's say, organisms. In this case, it was tomato juice who produced extracellular particles, and while we were isolating them from the tomato juice, we also found viruses inside. So they are the same size. You see down, it is the virus, which is like rod-like, let's say. There are many shapes of them, and all these structures are interconnected, and coming back to that, so we can see now that we can do it also artificially. So this is the liposomes. They're created from lecithin. It's edible. You can buy it in the market. I mean, you go to the store with healthy food, buy one box, it's lyophilized, and mix it with water and glycerol, and we produce these particles. So these are also the basis of the vaccines that we took for COVID. So it's a similar mechanism all over with that. So there are very recent images taken in Uppsala in Sweden of such liposomes carrying the cannabis cargo of Dr. Stuckel. And we can come to the next conclusion that these extracellular particles and liposomes are universal cargo carriers, or in Earth, and maybe they could also do it in space. So we were very happy to be invited to put some of our green friends, so I have a sample here, beautiful as well, to the spacecraft that's going to the space, and our mannequins will turn there and come back, hopefully, and we will see how it affected them. So we are a little bit worried because this takes so long now. They're in the box, in the dark, without light, without our support. You have to say hi to them, and you have to take into consideration how they feel. But we hope that they will come back. But if they do, they're really tough. Then we will say, wow, they could go who knows where. And we will analyze how they will do after. We have control samples as well, which are waiting in the same tubes here, and then we will compare how they feel. But this is just the first experiment, so we have never been thinking about space before. Now, with that, we are starting to think about it, and actually they're already there probably. maybe not this type, but some of them, because the atmosphere is continuous. So there are little particles in the atmosphere, and also in the end they had to go somewhere. So probably they went already. And we have also some technological ideas about the experiment. So research of extracellular vesicles is not so well developed yet. So methods have to be improved. In particular ones to, let's say, detect them and identify them. The images that we showed are probably one of the best available on that, but we don't know the composition. We don't know, while we are getting them, we are destroying the samples. So much has to be improved. And this is let's say, a nanospacer is an idea proposed recently by a gentleman from Netherlands. So it's very simple. You don't need fancy equipment. Now the cytometer to detect those particles costs like 200,000 euro, but this one is very cheap. It's only a piece of glass like that big, with the space between the upper one and the lower one so small that only small particles can get inside and cells remain outside. So then look at it on the normal microscope, optical, and it looks like that. So this is a cartoon still. But I have seen you can do. I mean, if nanoparticles get into that side on the left and the cells are left outside, you can actually count them then. So that is very suitable for the spacecraft because you don't have you don't want to have heavy equipment and expensive and sophisticated, which can break then because they are very sensitive, and then you have to filter the samples and all that. So such kind of simple ideas which actually cost not a lot of money and are, I think, very good. Yeah. So here we also cyanobacteria, not only microalgae, because they were the ones responsible to do this beautiful atmosphere for us. And the first idea is to support these, let's say, organisms to maybe do it again or do it better, or I don't know how. So we are playing here with very dangerous things, actually. We can plant up there something that will harm us in the end if we don't know what will come out. And the little particles, the extracellular vesicles, are quite powerful things. So we have to be careful even before think about it, not only reach for the stars, but really think about what we will do up there, because there are exponential laws behind that. So if they start to, let's say, multiply, it is so you have a little bottle like that. Next day you have 2 of them, and next day you have 4 of them, and you come very fast that you don't have a bowl big enough to feed them all. So space is perfect for them. They will have a lot of this. So other things will limit them. There will not be material available, maybe. There's limiting things, but they could be really powerful in expanding in space. And people are already building for the spacecraft the special reactors for them. Also, there are architectural ideas how to use them to people think that they will do fuel with them or eat them or colors for the clothes or many things that they could be used for. But we have to think about that their primary role is to create atmosphere, and I think they should be supported in this way, not to eat them or destroy them or do anything nasty to them. And so there is cyanobacteria, also very beautiful. And then now thinking about, this space, how these little creatures are very, very first beings that could be, let's say, comfortable in space and not with all the let's say, sacrifice and victims. Think about, many people who died and also the animals who were used, like this little dog, Laika, who went there and died. And therefore, the extension of the atmosphere, in fact, is like hundreds of the radii of the Earth, and we have to think about it. And support the life in this kind, where these extracellular particles could be one of the main players in the development. So thank you very much for your attention.
We thank you, Veronika. Do you have any questions, ladies and gentlemen? Any questions from the floor? Yes, there will be a question. Thank you very much. We will give you a microphone. Thanks.
Thank you, Veronika. It was very nice. And I would have two questions, actually. One, going backwards, algae. Have you considered genetically modifying circuiting genes, circuiting clock genes? Because the Mars, for example, has 24/6/5 hours compared to ours. So you would need to have a change in adaptation. And yep, that's my first question.
No, we have not considered to do anything to them, but support what we have. So our methods are mostly also to let's say, characterize them very gentle, as little interference with them as possible. But we, never really did much with the algae. We were after the extracellular vesicles mostly, and of course these things should be considered in the future.
Okay, I'll be interested in that for sure. The second question would be towards the EVs, the extracellular vesicles. Did you ever consider, and there's going to be a big jump, because that's a big jump from algae in a way, in my terms, into muscle development, and to go a little bit closer to the EVs, into their role in mitochondria, performance increase, and also in numbers and in terms of quality and quantity changes in mitochondria, because we know that there are quite a bit of signaling pathways and others, they play a key role in mitochondria. Do you do any on that?
Yes, of course.
Yeah.
So extracellular vesicles probably in all processes,
yeah,
biological can have a big role, and any knowledge on that's highly warranted in any of these fields. And I think in the future, these ideas of nanostructures, membranes, will have, so the knowledge on that will expand tremendously. So yes, of course, and they are interconnected, and also you can find connection between any field.
Yeah, that's correct. That's the beauty about biology, isn't it? That everything is connected. Just 2 meters away from you, there is the muscle,
exactly,
electrical muscle stimulation suit, just on the right, and that has a big effect on mitochondria quality and quantity, in terms of quality and quantity of mitochondria. And of course, we are interlinked in that manner, because your EVs would improve mitochondria as well.
Yes, or destroy. So
yeah,
you have to know. And I think, yes, it affects, it's ongoing now.
Yep.
In us.
Yes,
all of
every minute. Yeah.
Yes.
Okay, thank you. Brilliant.
Lovely stuff. Thank you very much. Thank you, Veronika, for answering these questions. For the first time today, I felt a real outsider. Anyway, thank you very much for answering the questions.
Thank you.
Can you show us the sample again from your
Yes,
pocket? I mean, that was not easy to enter the country at the border for Veronika, but she's here. Thank you.
I put it, nobody.
Yeah, some strange questions from the police at the border, but anyway, thank you very much, Veronika. You are a rock star. Thank you very much.